There is a furnace at the center of our solar system that has been burning for 4.5 billion years. Right now, at this very moment, in a region roughly 15 million degrees Celsius at its core, hydrogen nuclei are being slammed together with such ferocious force that they fuse, releasing energy of a magnitude that staggers the imagination. We call this process nuclear fusion, and it is the engine behind every star you have ever seen. The sun converts roughly 4 million metric tons of its own mass into pure energy every single second through the relationship Einstein captured so cleanly in 4 characters, E equals mc squared. That energy does not stay locked inside the star. It begins a journey, and at the end of that journey, it lands on you. A single photon of light, born from a nuclear collision deep in the solar core, takes somewhere between 10,000 and 100,000 years just to reach the surface of the sun. It bounces, gets absorbed, re-emitted, redirected in a tortured random walk through the dense plasma of the solar interior. But once it clears the photosphere and escapes into open space, everything changes. From that point, it travels at the only speed physics allows for massless particles, 299,792,458 meters per second. It crosses the 93 million miles between the sun and the surface of the Earth in precisely 8 minutes and 20 seconds. And then it does something that no laboratory experiment, no pharmaceutical compound, no engineered molecule has ever done with anything close to the same efficiency or elegance. It changes your brain. The word drug, in a precise pharmacological sense, describes any chemical agent that alters the structure or behavior of molecules in the nervous system, producing measurable changes in mood, perception, cognition, or physiology. We tend to picture drugs as pills, powders, or injected compounds. But this framing is too narrow. It privileges chemistry over physics. What matters, at the molecular level, is not how an agent arrives, but what it does once it interacts with the biological machinery inside you. And sunlight, pure electromagnetic radiation, interacts with that machinery in ways so deep and so pervasive that if you stripped it from human biology entirely, you would not merely feel tired or sad. The fundamental architecture of your circadian regulation, your hormonal balance, your neurotransmitter synthesis, and your immune calibration would collapse. Let us start at the photon itself, because understanding what light physically is turns out to be the foundation of everything that follows. Light is not a beam of tiny bullets traveling through space. It is an oscillating electromagnetic field, a coupled wave of electric and magnetic disturbance propagating outward from its source. Quantum mechanics adds a layer of complexity here that is essential. Light also comes in discrete packets, quanta of energy whose magnitude is determined by their frequency. High-frequency light carries more energy per photon. Low-frequency light carries less. Visible light, the narrow band your eye can detect, sits between roughly 400 and 700 nanometers in wavelength. Ultraviolet light sits just above that range in frequency, below it in wavelength, and carries enough energy per photon to do something visible light cannot do to biological molecules. It can break chemical bonds. Your eye is not a camera. That analogy, though common, deeply undersells the physics at work. The retina is a layered structure of extraordinary complexity, containing around 130 million photoreceptor cells, rods for low-light monochrome vision, and cones for color and daylight acuity. Inside the outer segment of each photoreceptor cell is a molecule called retinal, a derivative of vitamin A, embedded within a larger protein called opsin. Together they form the compound rhodopsin, and this molecule sits at the boundary between physics and neuroscience in a way that few biological structures do. When a photon of the right frequency strikes the retinal molecule, it does not merely warm it or vibrate it gently. The photon is absorbed, and its energy drives a specific quantum mechanical event. The retinal molecule undergoes photoisomerization. It physically changes shape. Retinal exists in the dark in what chemists call the 1,1 cis configuration. The molecule is kinked, bent at a particular carbon-carbon double bond. When the photon strikes, its energy causes that double bond to rotate, and within femtoseconds, retinal snaps from the 1,1 cis configuration into the all-trans configuration, the molecule straightens. This geometric change is one of the fastest photochemical reactions in all of biology. And that change in shape, that physical restructuring triggered by a packet of electromagnetic energy born in a nuclear furnace 93 million miles away, is the first event in the chain that produces vision. It is also the first event in the chain that eventually tells your brain what time of day it is. The straightening of retinal causes the opsin protein wrapped around it to change its own configuration. This activated form of rhodopsin then triggers what biologists call a signal transduction cascade, a rapid sequential activation of molecular switches, G proteins, enzymes, ion channels, each amplifying the signal at every step. A single photon, through this cascade, can produce the closure of hundreds of ion channels in the photoreceptor membrane. The electrical potential across that membrane shifts. A nerve impulse is generated. The signal races along the optic nerve toward the brain, and at this point, the information carried by that single photon has already been amplified by a factor of millions. The sun has sent you a message, and your cells have shouted it across your nervous system. Now consider where this message goes, because the routing of light information through the brain is not as simple as the pathway to the visual cortex. There is a separate channel, a distinct population of retinal cells, different from the rods and cones, discovered only in the early 2000s, contains its own photopigment called melanopsin. These are the intrinsically photosensitive retinal ganglion cells, and they have a peak sensitivity to short wavelength blue light around 480 nanometers. They do not contribute meaningfully to the images you see. What they do instead is project directly to a tiny structure in the hypothalamus called the suprachiasmatic nucleus, a cluster of roughly 20,000 neurons that functions as the master biological clock of the mammalian brain. This is the circadian pacemaker, and it is calibrated, reset, and synchronized primarily by the presence and intensity of light. The suprachiasmatic nucleus operates on a roughly 24-hour oscillation generated by interlocking feedback loops of gene expression. Proteins coded by genes with names like Clock and Bmal1 drive the expression of inhibitory proteins coded by period and cryptochrome genes. Those inhibitory proteins accumulate, shut down their own production, degrade, and allow the cycle to restart. This molecular clock runs in virtually every cell of your body, not just the suprachiasmatic nucleus. Liver cells, skin cells, immune cells, heart muscle cells all carry their own peripheral clocks. But the suprachiasmatic nucleus is the conductor. It receives the light signal from the melanopsin-containing retinal cells and uses that signal to synchronize all of those peripheral clocks to the actual rotation of the Earth. Sunlight, in this framing, is not a metaphor for biological timing. It is the physical mechanism by which your body keeps time. When morning light reaches those melanopsin-containing cells, the signal reaches the suprachiasmatic nucleus, which then sends outputs that cascade across the entire brain and body. One of the most consequential of these outputs runs through the hypothalamus to the pituitary gland and then to the adrenal cortex, triggering the release of cortisol. Cortisol has an undeserved reputation in popular culture as merely a stress hormone, something to be minimized. But the morning cortisol surge is a precisely engineered physiological event. It mobilizes glucose, activates metabolic pathways, raises core body temperature, enhances immune surveillance, and brings the brain from the diffuse, low-frequency oscillations of sleep into the high-frequency alertness of wakefulness. Without the light cue, this cortisol pulse blunts, delays, and loses its sharp amplitude. The machinery is still there, but the timing signal that coordinates it is absent. Serotonin synthesis follows a parallel pathway. The amino acid tryptophan, obtained from dietary protein, is converted in neurons to 5-hydroxytryptophan and then to serotonin, a neurotransmitter involved in mood regulation, social behavior, appetite, sleep, and the modulation of pain. What is less commonly understood outside of neuroscience is that light exposure, particularly broad-spectrum sunlight hitting the retina, directly modulates the rate of serotonin synthesis in the raphe nuclei of the brainstem. The precise mechanism involves the light-driven changes in firing rate of the retinal projection neurons that reach the raphe via the hypothalamus. When light intensity is high, serotonergic activity increases. The brain, quite literally, produces more of this molecule in response to photons from the sun. This is not a metaphor. This is measurable, reproducible photochemistry that depends on the specific electromagnetic spectrum of sunlight and the specific absorption properties of the biological molecules in your visual system. Melatonin occupies the other side of this equation, and understanding its physics reveals the extraordinary precision of the system. Melatonin is synthesized in the pineal gland, a small pine cone-shaped structure sitting near the center of the brain, from serotonin, under the instruction of the suprachiasmatic nucleus. Crucially, the suprachiasmatic nucleus suppresses melatonin synthesis in the presence of light and allows it to rise in darkness. The photoreceptive pathway from the retina to the suprachiasmatic nucleus to the pineal gland is so sensitive that even moderate indoor light, a fraction of the intensity of outdoor sunlight, can partially suppress melatonin. Outdoor sunlight at midday can reach 100,000 lux of illumination. A well-lit office interior reaches around 500 lux. The difference is 2 orders of magnitude, and that difference has measurable consequences for the depth and timing of the melatonin signal, the internal hormonal broadcast that tells your body it is night. Dopamine adds another dimension. The retina itself synthesizes and releases dopamine in a light-dependent manner. Retinal dopamine is involved in the adaptation of the eye to daylight conditions, the switching from rod-dominated to cone-dominated processing, and the regulation of eye growth during development. But this retinal dopaminergic system does not operate in isolation from the rest of the brain. The dopamine pathways that govern motivation, reward anticipation, motor control, and executive function all carry some degree of circadian modulation, and disrupting the light cue upstream distorts that modulation. People who work night shifts over many years, chronically exposed to light at the wrong phase of their biological clock, show measurable differences in dopaminergic tone compared to people whose light exposure aligns with their circadian phase. The alignment between photons and biology, between the physics of sunlight and the neurochemistry of motivation, is not incidental. It is structural. Now move beyond the retina entirely to the skin. The skin contains no photoreceptors in the classical sense. It cannot see, but it carries out photochemistry of its own. The most consequential of these reactions is the synthesis of vitamin D, which is not, despite its name, merely a dietary vitamin. It is a steroid hormone synthesized in a photochemical reaction that depends on ultraviolet B radiation, wavelengths between approximately 280 and 315 nanometers. When this UVB radiation penetrates the epidermis and reaches the deeper layers of the skin, it strikes a molecule called 7-dehydrocholesterol, a precursor molecule in the cholesterol synthesis pathway that resides in cell membranes throughout the skin. The UVB photon carries enough energy to break a specific chemical bond in the B ring of that molecule, opening the ring and forming previtamin D3. That previtamin then undergoes thermal isomerization, a heat-driven shape change rather than a photochemical one, to form vitamin D3. The liver then adds a hydroxyl group, the kidneys add another, and the result is the fully active hormonal form, calcitriol. Calcitriol does not function like a neurotransmitter, acting at a synapse over milliseconds. It acts as a nuclear hormone. It enters cells, crosses into the nucleus, and binds directly to the vitamin D receptor, a protein that then acts as a transcription factor, binding to specific sequences of DNA and modulating the expression of hundreds of genes. Current estimates place the number of genes regulated by the vitamin D receptor at somewhere between 2 and 3% of the entire human genome. These include genes involved in calcium absorption, immune cell differentiation, inflammatory signaling, cell proliferation, and neural development. The photon from the sun, through this pathway, is reaching into the nucleus of your cells and adjusting which parts of your genome are being read. The distance between that nuclear reaction in the sun and the regulation of your DNA is 93 million miles. The messenger is a quantum of electromagnetic energy. The mechanism is photochemistry followed by nuclear receptor biology. The skin does not merely make vitamin D under light. It also produces nitric oxide from photolabile nitric oxide stores in the epidermis when struck by UVA radiation, wavelengths between 315 and 400 nanometers. Nitric oxide is a gaseous signaling molecule that enters the bloodstream and causes vasodilation, the relaxation and widening of blood vessels. This produces a measurable drop in blood pressure. Studies comparing cardiovascular disease rates between populations at different latitudes have long noted a gradient that cannot be fully explained by dietary differences alone. The UVA-driven nitric oxide pathway is one plausible physical mechanism underlying part of that gradient. The sun, through a distinct photochemical pathway from the vitamin D synthesis route, is regulating the geometry of your circulatory system. Zoom out now and consider the evolutionary picture because it grounds all of this in the deepest physical context. Life on Earth evolved under this specific star with its specific electromagnetic emission spectrum for approximately 4 billion years. The sun emits radiation across a broad range of frequencies, but the atmosphere filters that radiation selectively. Water vapor, oxygen, and ozone absorb most ultraviolet below about 300 nanometers at the surface. The result is a characteristic surface spectrum dominated by visible light and near-infrared, with a narrow window of UVB allowed through. Biological systems did not evolve to tolerate this spectrum passively. They evolved to use it to build molecular machinery calibrated with extraordinary specificity to the frequencies that reliably penetrate to the surface. Retinal absorbs visible photons. Melanopsin preferentially absorbs blue wavelengths. 7. Dehydrocholesterol absorbs precisely the UVB wavelengths that reach the ground. This is not coincidence in any loose sense. It is the outcome of billions of years of selection pressure under a specific star with a specific spectrum. Your biochemistry is, in a precise physical sense, tuned to your local star. There is an irony buried in the history of medicine here that is worth noting. The 20th century produced extraordinary pharmaceutical tools, compounds designed by synthetic chemists to target specific receptors, enzymes, and ion channels with high affinity and selectivity. These compounds have prevented enormous amounts of suffering, but alongside that pharmacological revolution came a progressive decoupling of human biology from the environmental inputs it evolved to receive. Electric lighting allowed the manipulation of the light environment in ways that had no precedent in millions of years of primate evolution. People began spending the majority of their waking hours indoors, under artificial light sources with spectral profiles very different from sunlight, with intensities far below outdoor daylight, and crucially, with exposure at phases of the circadian cycle that the biological clock did not evolve to accommodate. The result is a population chronically exposed to the wrong light at the wrong times and insufficient light during the biologically critical morning window. The physics of this situation has not changed. The molecular machinery still waits for the signal. The melanopsin-containing retinal cells still project to the suprachiasmatic nucleus. The suprachiasmatic nucleus still coordinates the hormonal cascades, the cortisol pulse, the serotonin synthesis rate, the melatonin timing. The skin still converts UVB photons into a hormone that regulates hundreds of genes. But the input signal, the stream of photons that evolved biology expects to receive in the morning with a particular intensity and spectrum, has been progressively attenuated, shifted, and mistimed by the built environment. The circadian machinery runs on a degraded and misaligned light signal, and the downstream consequences ripple through mood regulation, immune function, metabolic timing, and sleep architecture in ways that epidemiological data have been documenting for decades. This is why the framing of sunlight as a drug is not rhetorical flourish. It is physically precise. A drug modulates the molecular machinery of the nervous system and body. Sunlight, through at least 4 distinct photochemical pathways, does exactly that, acting on receptors that evolution specifically built to receive this particular electromagnetic input, driving the synthesis of neuroactive compounds, modulating gene expression at the nuclear level, and synchronizing a distributed network of biological clocks to the geophysical reality of Earth rotating under its star. The difference between sunlight and a pharmaceutical compound is not that one affects biology and the other does not. The difference is that sunlight was present at the beginning, that life built itself around it, that the machinery inside you was calibrated to it over a time scale that makes all of human civilization look like a rounding error. Every morning, when photons that began their journey in nuclear collisions at the core of a star finally reach your retina, something ancient and precise happens. Molecules change shape, electrical signals propagate, hormones shift, clocks synchronize, and the machinery inside you, refined by billions of years of evolution under this particular star, continues doing exactly what it was built to do.